Well, first of all, thank you, Ian, Orit, and the rest of the organizers for inviting me. I'm excited to be here. I just arrived last night, so it's great to be here in person. I'm a biophysicist, so I'm going to tell you about the statistical physics, non-equilibrium statistical physics tools that we have been developing to try to understand self-organization in biological systems at many different scales. And first, I'm going to give you an overview about our philosophy, and then I will show you concrete examples of how we apply these tools, how we develop these tools to one particular system, of course, with the hope that these are universal tools that can be applied to many of these collective states and collective systems. So again, as I mentioned, I'm a biophysicist, and like many of my colleagues, we are fascinated by the fact that there is something different about being alive, and we would like to know what that is in the same way that we know what it is for a collection of atoms to be solid and for a collection of electrons to be superconducting. Now, if you look around, more or less immediately, you can identify things which are alive. And the criteria that we use in making this discrimination between the animate and inanimate world has nothing to do with the individual components, such as DNA or proteins. DNA or proteins are not alive. Being alive is a macroscopic state, while things like DNA and proteins are the components of the microscopic mechanisms by which this state is generated and maintained. And to illustrate this point, I'm going to talk about this beautiful video that you see on the slide here. This is actually a video taken in my group. First of all, this is an egg cell or oocyte of a sea star. It's a model system that we use in my group. And note that this is a very large cell. If you look at the scale bar, it's about 200 micron in diameter. And what you see is self-organized patterns of signaling proteins that orchestrate a very important process early in development, namely cell division. And of course, the ability to divide is a hallmark of life, at least life as we know it. Now, we may not know what these proteins are, and we can't see individual proteins either because of the scales. Protein is on the order of nanometer, and we're looking at micron to millimeter scale. But one thing we can all appreciate just watching this video, and that's the fact that we have this long wavelength patterns and processes that span the entire surface of the cell. They propagate from one side to the other side. They self-organize from one state to the other state. And this happens while the egg cell is continuously consuming ATP and dissipating energy. Other thing I want you to notice is that these patterns are very similar to a very famous reaction, BZ reaction. So oftentimes when I first show this video, some people just immediately start thinking about it. But there's a crucial difference between in these patterns in living systems and, of course, the type of patterns that you get in those chemical systems is the fact that these patterns have purpose. They actually are responsible for cell division, and that, I think, is a very important feature, that there is this kind of intimate relation between pattern and function in biology. Now, over the past, I think, many decades, molecular biology has been very successful in identifying the components, the parts, as well as microscopic mechanisms. However, I believe we have made less progress in identifying the features that are characteristic of this macroscopic living state. And I'm going to quote Ernest Everett Just, who I believe is a pioneering biologist in this area, where he says that the direct analysis of the state of being alive must never go below the order of organization which characterizes a life. And of course, if you think about this statement a little bit, you realize that this is actually at the heart of physics. One of the fundamental questions we ask in physics is not only discovering the underlying laws in the system, but in understanding and explaining the new laws that emerge when many individual components interact. And to discover these laws, we do not always keep track of all the electrons or protons. We're always looking for important variables, or what we call key degrees of freedom. And once these variables are identified, we combine them into what is called an order parameter, which is the quantification of the emergent phenomena. Now, in equilibrium physics, this concept of order parameter has been very powerful. And of course, it plays a crucial role in defining a wide range of phenomena. I think a classic example that we all remember from freshman physics is a magnetization. Now, asking for the order parameters of the living state is a very hard problem. It's also not a well-defined question. And this is because when equilibrium is given, statistical mechanics frames the behavior and phenomena in terms of energy minimization. Because these are closed systems, they don't exchange energy or matter with the environment. So we have this minimization principle. But when a system is out of equilibrium by necessity, which is the case in living systems, we can no longer follow an optimization principle. And the question is, can we identify general principles that can guide us in developing a physics of self-organization in living systems at multiple scales that can reach the same level of predictive power that has been the standard in any other area of physics? And of course, this is the challenge that we have set out to address under the umbrella of active matter physics. I'm showing this video. This is, of course, a flock of starlings. This is a kind of a more maximum intensity projection, so the image has memory of the birds. And I'm sure you have seen incredible videos from Ian here and others. But I think this is a classic example that we show. But I just want to kind of highlight the approach we take to try to understand this type of systems. So we develop experimental tools to observe and identify non-equilibrium degrees of freedom. We develop data analysis frameworks to define order parameter fields and to identify the broken symmetries in the system. And of course, very crucially, we develop theoretical frameworks which have predictive power. And of course, at this stage, the theory can give us that killer experiment which we can go back in the lab and try to see if we can realize the predictions. And this is kind of like a loop that generates really exciting, I think, results. Now, of course, our ultimate hope is that the concepts that we develop are system independent. I think this is at the heart of this universality that we love in physics. But we always need model systems with a rich phenomenology to develop these self-organization principles. And I'm going to quote Rob Phillips, who's a physicist in Caltech, where he says that we need hydrogen atoms because if you think about it, hydrogen atoms served as this quintessential test case and helped us understand the behavior of basically quantum mechanics all the way at the scale of a single atom to phenomena such as Bose-Einstein condensate. But we had this system which we could really, I guess, be able to understand these scales and also what happens when you have more of these components together. So the hydrogen atom of this talk is going to be the egg cell or oocyte of a C star. And this is, first of all, one of the oldest known developmental biology model systems. I'm referencing a paper from 1877. And one of the reasons that I think this is an incredible arena to understand self-organization is because multicellular development, as it has evolved over the past billion years, is really a marvel in these ideas of self-organization and information processing. You have to have many processes self-organized in space and time to give us robust outcomes, such as cell division. So let me just very briefly take you on a journey in space and time and to highlight some of the broken symmetries that we have identified and the emergent phenomena that we have discovered. And in the interest of time, I'm going to focus on one particular scale and show you how these approaches have been powerful in trying to understand collective behavior. So this is basically showing an axis that we go from the smallest scale at the intracellular scale all the way to organismal scale, where you have a collection of organisms. And I'm going to start with the smallest scale at the level of when we are at the intracellular scale, and talk about what's arguably the most important broken symmetry in non-equilibrium system and living system in particular, and that's time reversal. And my group has shown that fluctuations at the smallest scale encode thermodynamic arrow of time, and we have been developing tools from information theory and stochastic thermodynamics to identify spatiotemporal scales of dissipation. And just to give you a sense of what you're actually looking at, this is again that large cell that we are looking at. It's like about 200 micron, and all of these are organelles within the cell, and they're about a micron in size. We have many, many of them, and you're looking at kind of their fluctuations within this non-equilibrium environment. And in fact, we use the trajectories, the positional information from these millions of trajectories that we can extract, and then we can construct information theory metric to actually quantify dissipation in this type of systems. We have also kind of extended this to actually the patterns that I showed you using machine learning tools. I'll be happy to talk about that, but I will get back to this idea of a time reversal symmetry breaking, because it is really at the heart of all the processes that we observe in biology.
Is that supposed to be, so I'm kind of permuting my particles and looking at a mutual information between
two probability distributions, yes, yeah. And in this case, it would be the probability distribution of observing a forward process and a backward process, and that's kind of where the arrow of time basically comes in. And by the way, please feel free to interrupt me at any time. I'd rather be kind of this to be a more interactive talk.
And so x1 through xn are kind of equally spaced in time, like the state at equally spaced time intervals.
Right, so it's a path probability,
yeah,
yes. Yes, so that's I guess small scale and kind of the most important broken symmetry. I'm going to move up the scale as well as complexity, and we have been looking at the cellular scale, the patterns that I have shown you, and we've been focusing quite a bit on trying to understand how these biochemical signaling proteins organize at cellular scale. These are examples of some of these protein patterns that you observe. We're kind of squished the cell, so that's why now they look a bit elongated. And what we have shown is that actually cells use this type of patterns and different types of modules. So these are reaction diffusion type patterns, gradients. Cells use these modules and combine these modules to perform computational operations. For instance, the cell needs to find its center. It needs to sense its shape. And in fact, these patterns, the wavelength they generate, is a mechanism for them to be able to do this. And so we've been focusing quite a bit on this, and there are many other interesting features that you kind of like when you think about it from a non-equilibrium stat-mech perspective, you can look at these patterns, and basically if you look at the phase of the oscillation of the patterns, you see topological defects. So there's a range of different things that you can try to understand from a physics perspective in these systems. But also from a biological perspective, one thing to note is that it looks like I have a ladder of the patterns, and you can notice that as we go from top my right to the bottom left here, there seemed to be a change in the wavelength of the pattern, the chaotic kind of nature of the pattern. And in fact, it has something to do with the amount of dissipation within the cell. So if you need more complex patterns, you need to increase the ATP consumption or dissipation within these systems. I won't talk further about this, but happy to answer questions if you are interested in learning more. And the last thing I would say is that, of course, these patterns are kind of abundant in biology. They have certain purpose, but I think one of the important things about living systems is the fact that we have actually control over these type of systems, and we've been spending quite a bit of time trying to understand whether we can actually generate, once we know the phase space of these patterns, whether we can use tools from biology to be able to generate these patterns patterns on demand. So this is an example where we use light. So we use optogenetic tools to actually generate these patterns just by shining light on the cell. And that actually is related to how the cell shape deformation comes about. So we can engineer different types of cell shape. And this is a paper that's going to come out soon. And so there's this kind of notion of control as well, which we actually, by living systems, give us the possibility to do that. Okay, so this is spatiotemporal symmetry breaking. But today I'm going to focus on an area that we've been very excited about, and that's looking at chiral symmetry breaking, chirality in the dynamics. I'm going to go into details of that. And I'm going to talk about a collective state that emerges when you don't have a single egg cell, but you actually have a collection of these embryos coming together, and they form a collective state. And you get some very interesting features from this collective behavior. And I'm going to use this as a test case to, again, show you how we do experiments, how we do data analysis, and how we develop theoretical models and tools to be able to understand the dynamics within these systems. So for me personally, it's a very intriguing system, but I guess what I hope you also kind of get from this is that these tools are really universal and can be applied to any type of collective behavior that we observe on different scales. Okay, so I mentioned the hydrogen atom of the talk. I'm actually going to show you the periodic table of this talk. And what you're looking at is a branch of the phylogenetic tree. This is called the echinoderm branch. We are in California, so I think actually we do get this species from right around the coast. We have a diver, we call him, he goes and he dives and gets us this species. So echinoderms are this exceptional group of organisms where they start from a bilaterally symmetric larvae. So that's looking at those kind of schematics there. And then they develop into pentameral adult symmetry. And this symmetry breaking on its own is a truly fascinating concept. But what I'm going to focus today is when they're at this very early stages in development, so a bilaterally symmetric object. And we're going to look at their dynamics, the individual dynamics, and then what happens when you put a large number of them together. And you can see some aspects of it, which seems like they can come together to form this kind of collective state. And the most part of the talk will be focused on this Petiria miniata. This is the bat star, but this is a very robust, actually, process. You can look at many other species in this branch. They do the same thing. Some of them do even more interesting things. This is an example of sea urchin. Sea urchin starts from a bilaterally symmetric object and then slowly breaks shape symmetry as well. So at some point, it becomes more like a Eiffel Tower, a half Eiffel Tower structure. And then instead of forming actually kind of like a more crystal structure, it forms a liquid, an active liquid state. And you can get interesting vortex behavior and features there. And I guess the main message is that when we have many broken symmetries in living systems, and in particular, when you have time reversal symmetry breaking, which is the non-equilibriumness of these systems, different mechanisms of breaking chiral symmetry, and as well as a very important broken symmetry that I'm going to introduce, this action-reaction symmetry, which is basically we know very well from Newton's third law. Actually, new material properties, new features in this collective states emerges. Yes?
Question, is this adaptive? Like, do they do this for a reason?
Yeah, that's a very interesting question. We don't do anything kind of like artificial to them for this to form. And we just increase the density and they come together. They haven't been reported in nature, but they kind of like, we have some hypotheses of why a state like this, because people haven't really looked, but they have some hypotheses of why states like this could form. And I'm going to jump way ahead, but I'll give you our kind of like, favorite hypothesis at the moment, which is, as you will see later, when these kind of like they come together, they can actually generate work on the environment. And we believe that that could be actually a mechanism to change the temperature of the environment. And usually these objects, these kind of animals, they're broadcast spawners. They live in intertidal zones where you have a lot of fluctuations in density and water salinity, pH, temperature. And so that's currently one of our main hypotheses. I'll say one more thing about that. There also has been very recent work where they have shown that if you put a group of embryos, not of this type, but Xenopus frogs together, they have a higher chance of survival. And they showed this basically by generating a wound in one of these embryos. And usually when you generate a wound in a tissue, you get calcium signaling within that tissue. But it turns out that you actually get calcium signaling propagating through the whole collective. So it may very well be an allele effect as well. But yes, this is just interesting questions. Yeah. Yes?
I wonder if it could also be a way to increase oxygenation.
Yeah, so they do come to the water interface to actually do that. And what I will show you is the ones that are, this is a small cluster. But we have now seen that so hydrodynamic brings them together, and the ones that are in the middle, I mean, they actually have to rotate to continuously create like the force that's needed to bring them together. The ones that are in the middle, actually, they have to generate less power. So it may also be, and at this stage, they're not eating, so they have to conserve whatever nutrients that they have. So maybe that's also another kind of way to think about this. Okay, so all right, so what are these call them particles, but what are these organisms that we are looking at? So I'm going to start with this video to show you how we start the experiment. So each of these is one of these single oocytes. We fertilize the oocytes, and this is basically looking at the fertilization process. This is just a beautiful video of biology in action. You see that basically you have a fertilization envelope. And then the first cell division, symmetric cell division happens. And then this is followed by many, many events. And again, those patterns that I showed you are actually responsible for this cell division happening wherever it has to happen. And then at some point, you can see that we have many, many cells, millions of cells within this fertilization envelope. And if you notice, there is a layer on the outside. And this layer, the cells in this layer, they have a very important organ known as cilia. We're going to talk about this in detail, but this is basically an active polymer that comes out of the surface of the cell. It can actively beat. And when they beat asynchronously, you can see this washing machine behavior, just going back and forth. At some point, they synchronize, and then basically this is when these embryos release themselves from the fertilization envelope and basically start swimming in the wild. Now, this is kind of like looking at the dynamics at kind of like a coarse-grained level. I'm going to focus kind of like at the properties of the embryo itself. So you see where this type of dynamics actually originates from. So this is one of our embryos, and as you can see, it's slightly elongated along an axis of symmetry, which is called anterior-posterior axis. This is just the body axis of the animal. And what you can see is that basically these are kind of like cells that have a cilia. So all these hairy structures are this hairy cilia or organism that cilia. I guess that I mentioned. And you can see that basically you have an ellipsoidal object with many, many of these cilia on the surface. And now these cilia, they can beat. And in fact, they beat synchronously. And the beating pattern is responsible for the kind of like the actually breaking of left-right symmetry in many organisms, including us. Now, if you look closely at the box, the red box that I had, so just so you get an idea about basically the ciliary dynamics, again, this is the surface. You have many of these cells here. You can see that the cilia sticks out of the surface into the seawater. And then you can clearly see this very nice kind of like wave pattern, active wave pattern. And you can also see the synchronization that occurs between many of them. And this is very much, I guess, reminiscent of metachronal waves in stadiums. And I think what's really, truly amazing is that we're, again, like we think quite a bit about scales. And this is self-organization at the small, kind of like at a mesoscopic scale. And then, of course, this self-organization results in the fact, basically the process of them being able to push the fluid next to the surface and be able to swim. Now, because there is kind of like this kind of wave pattern induced by this ciliary beating, these objects swim in a particular fashion. So they don't swim forward, they actually swim forward and also rotate. So they have kind of like a corkscrew type motion. And that's why we call them chiral particles, because the chirality doesn't come in the structure, the chirality rather comes in their dynamics. And to kind of illustrate this better, we're going to, again, focus a little out to be able to see their dynamics within seawater. So just to orient ourselves, that's our ellipsoidal embryo. And what we're looking at is the water-air interface. The particles you see in the bottom are the actual embryos. The top is just a reflection at the water-air interface. And what you immediately observe is, I want you to focus on that object over there. They swim in the bulk, and once in a while they can come to the water-air interface, and they can attain a stable configuration such that the AP axis is perpendicular to the water-air interface. And in this configuration, they behave like spins. So if you think from a physics perspective, if you look from the top, they're basically spins. There's another way that they can break this chiral symmetry, which is, you can see that if you look at the one next to it, and that's when they come to the water-air interface, they actually have a finite angle with respect to the water-air interface, and then they precess about this axis of rotation. And this actually results in two mechanisms of breaking chiral symmetry. Having spins, so spinning of individual embryos, and I'm going to show you that actually results into a very interesting mechanical property, which is called autoelasticity. And then there is this mode of kind of orbiting, which is, again, if you are a physicist, physicists think a lot about spin-orbit type coupling. This precision of axis of rotation actually gives rise to another type of mechanical behavior in these systems. It also, I will show you how it gives rise to oscillations over very long, kind of large distances, or actually the entire collective. So these are the two mechanisms of chiral symmetry breaking we have within this system. Okay, so this is the individual properties of our particles. And of course, we're physicists, so I've always been fascinated by TP tops. So what do we do when we have a TP top? We just put a large number of them together. And before I play this video, let's orient ourselves. So now, instead of looking from the side, we're going to look from the top at the water-air interface. Each of these are one of these stable embryos at the water-air interface. And I'm going to play the video, and you'll see what happens in a bit. And what you see is basically this incredible phenomena whereby you get a spontaneous crystallization of a large assembly of these spinning starfish embryos. And over the course of their development, so first of all, note that this is a living system, so the development is progressing. This is happening over days. Over the course of development, they first form the crystal. They actually undergo this dynamics where the whole crystal breaks chiral symmetry and starts rotating. And then at the end, you see that noise gets introduced within the system. We can briefly talk about that. And then the whole system actually dissolves. And this is a very kind of like, it's actually a phenomena that you can observe by eye. This is my scale bar here, a millimeter. And just to give you a sense of scale, I'll show you this image. So this is our attempt on a shot on iPhone. This is basically a 6-well plate, if it means anything to you. If not, this is basically a few centimeters across. and a couple of centimeters of water within. And then you can see initial kind of like nucleation of these objects coming to the surface, and then growth, and ultimately they would just dissolve. So I'm going to just spend a little bit of time, and if you're interested, we can go more into detail, just to give you an idea of why actually this happens. So it turns out that hydrodynamic is the main mechanism by which these objects become attractive particles. Yes?
Sorry, Nikta, could I just slow you down a bit? I'm trying to kind of reconcile these biophysical events within the kind of life cycle of the organization.
Yeah.
Could you just kind of maybe, you may have said it already, may have gone by, but like how do they fit together, the kind of phenomena that can scale?
So this is looking at, so the kind of the time that we use is called post-fertilization. So this is the time T0 is where we kind of like, we start the fertilization, and then you get these objects, and kind of like initially, and then this whole process, what we observe is over a couple of days. And this is very early stages in development, even before they kind of like become kind of like start developing their whole complicated structures. So the whole process really happens in kind of like on the order of a few days, still quite a bit for like just having a steady state. But over this course of development, what happens is that the only thing that changes, for the most part, I usually put a slide there, but I somehow decided to take that out. But the embryos become just slightly elongated, and that's kind of the main feature that changes from kind of like time point to time point. Because of that, they become less stable at the water interface, because now they have kind of like this stability angle. You can actually do calculation to know what exactly that stability angle is. So they become less stable. And then the cilia initially is really homogeneously distributed along the surface. And over the time, they're still homogeneously distributed, but the flow patterns start changing slightly. And of course, as I'm able to briefly talk about this, hydrodynamic is the main mechanism by which this come together. And that's, again, another reason that the whole crystal starts dissolving. But the shape, by all means, still ellipsoidal, and kind of like you still get this Stokes slit type singularity, which is responsible for kind of like for them to come together. Okay, so yes.
Are the properties of liquid change from the beginning to the end?
Properties of the liquid, no. So we're looking at, we basically have kind of like a very large dish. So it's seawater within, and kind of like it's, there's nothing in the water that kind of like we...
Do they interact with environment?
They do, and not kind of, so they, in principle, we are not changing anything in the environment, but in fact, they do interact with the environment, and we're now beginning to see, I will maybe at the end show you something. We're now beginning to see that, I mentioned that they all have the same handedness, but we have now reasons to believe that they're actually bistable particles, whereas like, whereby they prefer a certain direction to rotate, but then depending on the strain or some kind of environmental cue, they can actually switch that kind of like
But I mean,
rotation.
water can dissolve all kinds of things. Maybe
Right.
they release something and the viscosity changes and things like that.
From what we have seen, that hasn't been really an effect that's kind of like noticeable, at least. But I will show you how this idea that they change, like environment can have effect on the behavior is definitely something we are exploring, but not through water. Like we're actually introducing kind of like boundaries and kind of like geometrical, constraints, and that actually shows that already they can change their dynamics in response to that. Okay, so maybe I'll just quickly go over this. So basically, this is the main reason that kind of like these particles are become attractive is the fact that if you look from the top, so you have cilia beating, and what you notice is this is a PIV image. Basically, we put fluorescent beads in water, and then we image the dynamics of those so we can get the flow lines, the stream lines. And then you can see that basically as the embryo rotates, so spin as it rotates, it's pulling the fluid inward, and then it's pushing it outward. And that basically is, you can show that this can be well understood in terms of Stokes-Lett equation, and when you have kind of like a generic fluid flow around a force-generating object. And this Stokes-Lett singularities seems to be enough to at least describe the kind of dynamics of single, double, and triplet objects. And again, happy to go into more details of that, but I'm going to maybe move on. And one thing I want to note is that so the Stokes-Lett is going to create basically the hydrodynamic, long-range hydrodynamic attraction. So if you look at the two-body problem, you can see that it attracts the embryos together. You, of course, have steric repulsion within this system. But if you note, when they come together, because of the fact that they spin, at this interface, you have both hydrodynamic lubrication, of course, interaction, but you also have this very long cilia that start interacting with each other. And that is actually a source of getting a transverse force and torque within the system. And that's noticeable because you can see that they come together and then they spin with respect to each other. You can have also instances where you also get a transverse force, kind of like additional force within the system, and that's when the center of the mass can move as well. And the nature of these forces are non-reciprocal, and that's kind of like the feature I was mentioning beginning in the talk. So these are basically, they defy this action-reaction symmetry, so they break action and reaction symmetry, and this is what we call a microscopic non-reciprocal interaction within the system. And of course, a combination of these interactions, reciprocal and non-reciprocal, gives rise to a range of interesting behavior that we see in the system. The first obvious one being the fact that you basically get a global rotation or global chirosymmetry breaking within the system. By the way, how much? I just go all the way to.
You're good because okay. I'll accumulate the time where people ask questions. So just keep.
All right. Okay, then I keep going. Yes.
I'll give you a warning.
Yes. Okay. Perfect. Yes. So again, I'm going to show this because I want to kind of like demonstrate how we can actually do a lot of, we can use this ideas from physics and kind of crystals to quantify the dynamics and also structure within these systems. So this is one example where we wanted to look at this disorder to order to disorder transition. And we do so by introducing an order parameter, which is an orientational order parameter. This is one example of the order parameter you get. These are experimental videos. And basically what we are looking at is a local hexagonal order, and that's looking at the magnitude of this psi 6, this order parameter that you can see. And then you can also look at the local bond orientation, which is the phase of psi 6. And you can see that initially you have all these crystallites, like you have multiple nucleation events. These crystallites come together, so you have a polycrystallite object. And so you have a lot of defects, and kind of like that's evident from the magnitude and phase of the psi 6. But because these domains can rotate with respect to each other, in intermediate times you end up with a nearly defect-free crystal. And then towards the end, if you notice, all of a sudden you start getting a lot of defects within the system, both in the center but also on the boundary. And that's, again, because of the fact that the shape of the particle is changing, so they're less stable. And then also the fact that they can kind of like move away from this. So that's, again, the mechanism by which they dissolve ultimately. So this is kind of like looking at a crystal and its ground state. But we started observing something very interesting when we looked at the excited state in this system. And in particular, what I'm going to show you is if we move from the lab reference frame to the co-rotating frame of the crystal, oftentimes we would observe phenomena like this. So again, this is the co-rotating frame of the crystal. And what you see is basically we have shear cycles and oscillations that span the entire surface of this crystal. They seem to go around in a chiral fashion. And this is a very surprising phenomena. And if you're not surprised, let me remind you that this is an overdamped system. And the presence of such kind of like long-lived oscillations, which is on the order of hour, is truly comes from the non-equilibrium nature of the system, and I'm going to briefly mention that. And the kind of like at the heart of this type of oscillations is the fact that we have both reciprocal type interactions, microscopic reciprocal interactions, but also this microscopic non-reciprocal interactions. And you will see how in the next slide how these microscopic non-reciprocity can actually emerge at the much larger scale as a macroscopic non-reciprocal response in this type of systems. And of course, this can open up a range of different possibilities.
Sorry, Nikta, I'm very confused about the energetics here. So you've got the Stokes flow, so you've got all the energy loss within the fluid. Where's the energy pumped? Like, where's it coming from?
It's a living system, so it's continuously, yes. So that's actually the whole the kind of like the point, yes. So they're kind of continuously kind of like consuming ATP, doing dissipation to overcome this dissipative losses, and yes. I'll show you actually, it's basically you're building an engine, thermodynamic engine using this, which is quite, I find it really mind-blowing. Okay, I guess just to kind of like, just to show you how the nature of these kind of like displacement strain oscillations. This is again using kind of like looking at the displacement field, looking at the angle, and I think it gives a better visual of how this kind of like collective behavior looks like at this scale. Now, I'm going to maybe, if you have never dealt with mechanical systems or overdamped mechanical systems, I'm just going to quickly walk you through why to us this was an incredible discovery and why it has opened up a possibility of thinking about designing mechanical systems. So kind of, this is standard elasticity, textbook elasticity. This is basically Hooke's law, F equals kx. I'm just writing k in this tensorial format. And in standard elasticity, we derive the entries of stiffness tensor under a variety of assumptions. And one of the very important assumptions that have this important consequences is energy conservation. So you have this energy conservation in equilibrium systems, and that actually results in basically the entries of this stiffness and tensor to be limited to two diagonal entries. One is the bulk moduli, and the other one is the shear moduli. And again, this comes about because of the fact that you have this energy minimization principle within these systems. Now, in 2020, there was this beautiful work from Vincenzo Vitelli and co-workers in University of Chicago, where they showed that if you relax this energy conservation constraint, one can unravel unexpected mechanical behavior that previously been overlooked. And this is because entirely new moduli appear in the stiffness tensor. And these are off-diagonal components. And this is just because, again, we relax that constraint. And now, of course, you can see that these are off-diagonal components. So in practice, they can couple different modes of stress and strain within the system. And in particular, now you can actually have a material where its response is actually non-reciprocal. And the way to think about it is that you can actually have a material, when you push on it, it actually rotates. And that usually it's impossible to realize in an equilibrium setting. And again, this kind of like macroscopic non-reciprocal response originates from the fact that you have this microscopic non-reciprocity at the small scale. You can actually start from the equations that describe the hydrodynamic equations that describe the dynamics at this scale, coarse-grained, and you can see how you can back out this I guess, a continuum theory at the much larger scale. And one of the consequences of, and they term this odd elasticity because you have this off-diagonal components. And one of the consequences of odd elasticity is the fact that you can actually, under certain excitations of the system, you can get oscillations in the principal components of the displacement gradient tensor, namely divergence, curl, shear 1, and shear 2. And to illustrate this, these are experimental data. We extract these components, and what you're looking at is basically going around the boundary of the crystal and plotting the magnitude of these components as a function of time, and that actually, it's a kymograph. It clearly gives you the notion of the oscillation within this system. But one thing to note is that you can see that they actually decay, and of course the decay comes from the fact that you have these other moduli, bulk and shear moduli, so you're still in an overdamped system, so this type of oscillations eventually decay, but still it's quite long-lived for overdamped system. And what's kind of quite amusing is why should we be excited about this type of oscillations? And to kind of like, I guess, convey my excitement to you, I'm going to do one thing. I'm going to take two of these components, shear 1 and shear 2. And what I'm going to do is I'm going to span a phase space. I'm going to construct a phase space spanned by these two components, shear 1 and shear 2. And what you can see is that basically the trajectories within phase space is tracing a closed ellipse. And this ellipse basically is an indication of an emergence of an autonomous, self-sustaining, elastic engine cycle in which the system is converting internal energy into mechanical work to offset these dissipative losses. And in fact, the area that's embedded by these ellipse is the work that's done on the environment. So if you do the science to make sure that it's the correct thing, is basically you get the work that's being done on the environment. And I think what's quite amusing is, again, using rigid body mechanics, you have built a system that's a thermodynamic engine. And we've been focusing a lot about under kind of like trying to measure dissipation from this type of trajectories in phase space. I'll skip that, but kind of like, again, you can use information theory to get actually the bounds of the dissipation within the system. And you can see when the crystal gets excited, you are at the height of kind of like energy dissipation, work, entropy production within the system. And then, of course, eventually, it just goes back to its kind of like original state. Okay, so this was looking at what happens when you have spins within your system. I'm going to show you what happens when you have this other mode of chiral symmetry breaking. And just to kind of remind you, we could have a particle that can be either in either of these two states, either be a spin or orbit, or precess about this axis of rotation. And this type of behavior, actually, if you look at the bulk, and this is a maximum intensity projection of these particles, you can see that they either kind of like rotate their body, but they go straight, or they kind of like have this kind of like curved linear type motion. And this is kind of like correlates with this angle of that they make at the water-air interface, so this rectilinear or helical type motion. Now, the question we had was what happens if the particle can switch from one state to the other state? And this is basically the type of phenomena that we observed. Again, this is a very large crystal. And then what you observe is that initially we have a crystal that just has fluctuations. But at some point, you can see that it switches from just fluctuations to synchronized oscillations over very, very large distances, again, like the entire surface of the crystal. The nature of these oscillations are very different from the odd elasticity, the ones that originate from odd elasticity. And to see that better, first of all, these are velocity oscillations. So we're going to look at velocity as a parameter. So what you're looking at here is kind of a center within the crystal, and we're looking at the fraction that are oscillating and fraction that are fluctuating, and the arrow is showing you the velocity. And you can clearly see that the system, this is just average over Vx and Vy. The system initially just fluctuates, and then at some point it switches to a different state, And this is equal amplitude oscillations over, again, many periods, and then eventually relaxes back to its kind of oscillation state, to its fluctuating state. So this is basically, we have a two-state system, and this two-state system can switch go from one to the other one. And again, the velocity oscillations here comes about in, the oscillations you see are the velocity oscillations. And a long story short, the way we understand this, I'm just going to quickly show the equations that describe the system and where this type of oscillations come from. So we have kind of like a set of equations that this continuum equations that describe the behavior of the system. We have displacement fields, which is basically has linear elasticity components, which can have odd and even elasticity. And then we have these additional terms, which actually come about because of the fact that the embryos can kind of tilt. And when they tilt, they actually end up with kind of like an orientation-dependent self-propulsion, which is a new way of introducing activity within the system. And this orientation-dependent self-propulsion then can kind of like create the type of dynamics we see in the system. And you can write down other equations that describe basically the angle of your velocity in the xy direction. So it gives you the azimuthal angle. And then this theta, which is the angle that it makes with the water-air interface. And this is a phenomenological equation. We have two equations. One is theta 0, the other one is theta naught. And then we basically kind of like describe the system as being bistable in this tilt angle. And for those of you who think about populations, this is basically kind of like comes from population dynamics. So you have kind of a region that under certain parameters, it can grow and then span the entire surface and then eventually shrink. And we have kind of evidence of like how these come about. We have some theory predicts some couplings between the oscillations in the strain field as well as displacement field. I don't know if I have, I guess, yeah, I think I have a slide on that. But I guess the gist of it is that a linear odd elasticity and this tilt and precession dynamics can describe the behavior of the system. And I'm just going to show you, this is predictions from the theory, which we're actually looking at in the experiment. This is getting tilt angle from experimental data. This is looking at the divergence, and this is looking at the omega, the rotation frequency. And then we get basically this in-phase oscillations when the system is oscillating. Both the theta and theta, of course, is at a higher angle, and it also oscillates. And then we get this in-phase coupling between the divergence and omega when the system is basically in the oscillatory state. Now, very initially in the talk, I mentioned that biology gives you this opportunity to control the system. And we were, for the longest time, thinking whether we can actually push the system from one state to the other state. And I'm going to show you the experimental, protocols that we have developed to be able to do that. And this is one example. So what you're looking at, I'm going to call this a crystal in a box experiment. So we confine our crystal to a U-shaped chamber with one edge that we can control with a piston. We can either apply a uniaxial compression or we can actually apply oscillatory compression. And in fact, what we can see is that when we apply certain compression rates, we can actually excite the system to go from one state to the other state. And this is repeatable, so you can kind of like bring the piston back and then repeat this many, many times. And it turns out that not all, we have a Goldilocks situation, so not all compression strain rates are created equal. And you can see that here. So this is plotting compression strain rate as a function of the total oscillation duration. And if you are too kind of like in the low compression strain rates, what we are doing is we are just inducing low wave vector deformations within the system. at high compression rates, we're actually introducing plastic deformations within the system, and it is in this region where we actually get nice long-lived oscillation within the system. So this is kind of very exciting. Again, the system is exhibiting two types of macroscopic behavior. One, odd elasticity. The other one is gyroelasticity, because it has this kind of notion of orbit and precession. For the longest time, we were hoping to be able to understand the contributions of all these features to the dynamics of the system. This is a collaboration that I have had with my colleague Jorn Dankel for many, many years, and this is a result that we are very excited about. And again, I'm happy to go into the details, but I want to mostly talk about the result. So Jorn and his group, they developed this dynamic mode decomposition technique so that we can actually, from the data, extract the prominent or important modes within the system. So I'm going to show you only the first 9 modes, so a low rank representation of the system. And this is basically the embryo trajectories. And this is the reconstructed trajectories using 9-mode DMD. And then you can see that it basically decomposes into 3 important modes. The first one is the Goldstone mode, which is this global rotation that we get in the system. Then we have about, I guess, 7 modes that are going to give you acoustic modes, which are very reminiscent of vibrations that you get within a crystal. And then there are 2 modes that are going to give you this circular type, rotation. And these are actually reminiscent of optical modes in condensed matter type systems. And the nature of these is basically from the spin dynamics is going to give you the global rotation and the vibrations. And it's basically this precision of the axis of rotation is going to give you this optical modes. Now, this part is kind of like very, if you are a physicist, you know what we're going to do next. We're going to put this on a dispersion relation so we understand kind of like the dynamics better. And you can kind of immediately see that this is in the oscillating state. This is in the fluctuating state. So the 7 modes that I did, we don't have the Goldstone mode here. But the 7 modes that are responsible for the vibrations, these are going to give you acoustic branch. You actually get this very nice quadratic dependence, which actually comes from the fact that you have odd elasticity within your system. And then you get these 2 other modes, which is going to give you a flat band. Right now, it's just 2 modes, but we actually have much longer. We have a lot of other modes, so it's really a band. But that flat band is actually a dissipationless band that occurs in the system when the system switches from one state to the other state. And I'm going to kind of like, you can do a lot of different things, can measure odd elasticity like this. But what I'm really excited about is basically the idea that this type of mode decomposition is going to open up the possibility to think about both non-equilibriumness within this system, but also non-linearities within this system. Because if you have a non-equilibrium linear systems, the modes should not couple. There are independent degrees of freedom. But when you start looking at spatial correlations or anti-symmetrized time correlations, you actually start seeing this checkerboard type patterns that emerge. And again, going back to this idea of non-equilibrium, measuring non-equilibrium and dissipation in the system, we have developed tools to really understand basically where these couplings come from and what's basically their contributions to the dissipation within the system. And I'm going to give you one example. And so, for instance, you can look at you see that there's a correlation between these 2 modes. So you can reconstruct your dynamics using these modes. And then what you can basically get is looking at the divergence of the reconstructed field and the curl of the reconstructed strain of the reconstructed field. And you get this really nice cycles or the velocity. And then you can actually see that in odd elasticity, you get strain cycles. In gyroelasticity, you actually only get velocity cycles, but not strain cycles. So again, this is really now a blueprint for thinking about how to design materials and how to extract work from these systems. And with that, I'm just going to show a couple of just beautiful videos and with how we are thinking about these systems in general. The first, I guess, direction that I will talk about is everything I told you is basically starting from the same particles, which are at the same developmental time. But turns out that you can play this game of actually mixing them at different developmental time, and then you can start looking at the population dynamics within kind of like collections that where you tune the non-reciprocity between these particles. And you can get many types of phase behavior. You can, depending on the configuration, initial condition. This is an example where if we add sequentially, and you can see that basically you have a phase coexistence. But if you mix them together and you add them together, you actually go from, this is experiments, you go from a chiral flock into a chiral crystal state. And we're kind of like thinking about how to understand the dynamics of this. And then I guess lastly, I'll show you this, not just a starfish thing. This is sea urchin, and you can kind of now see that they have a very unusual shape. So when you look at them, kind of like when they make a bound state, they're no longer kind of like a bound state which rotate with respect to each other. They actually have alignment interactions as well. And this alignment interaction then of course, gives rise to basically things that are much more similar to flocking behavior that we see in many other examples. And of course, the ultimate goal is to be able to design this. So we have started this collaboration with Metin Siti's group, where they have this kind of magnetic disks, and they can start basically actuating them from using an external field, and then see if we can emulate some of these behavior. Of course, the crucial difference being that these are not autonomous. I mean, living systems are autonomous. These are, of course, driven from the boundary, but maybe there are interesting features. And promise the last thing I want to say is because a direction that I'm very excited about, and this is the idea that, so I come from active matter field, and active matter for the longest time has been focusing on how you go from interactions between the individuals to an emergent property at the group scale. And that's kind of like, it has been very successful in describing this transition. But biological systems have this other transition whereby you go from a kind of like a group of organism or a group of cells or a group of agents to actually an individual, because that individual is fulfilling a purpose. And I think this is a very interesting transition that really opens up the possibility of trying to apply some of these kind of active matter principles, but thinking about how this happens. And I'm kind of like, this is something I've been thinking about, and we have found a very interesting model system to try to explore this. So this is a sea sponge. This is my student Bill Crockett. So there was some beautiful experiments from Wilson in 1910 where he showed that you can get a piece of sponge, you can put it in calcium, magnesium-free seawater, the cells dissociate, you have only 3 cell types, they de-differentiate, they become stem cells, and then you put them back together, you add calcium and magnesium, and they start actually coalescing and forming kind of like intricate structures. And at some point, of course, then this is at the kind of like heart of how you get this multicellularity and the onset of this transition. So I'm going to show you some very preliminary experiments. Again, these are experiments. They're not just simulations of phase separation. So this is looking at a very large dish. We just added a homogeneous solution of cells inside, and then kind of like within a few minutes, you start seeing basically kind of like this spinodal decomposition happening. And then as the time progresses, you also see kind of elongation of these regions and then a network structure happening. So I'm really excited because this these ideas of how kind of thermodynamics could put bound on where like this transition of like just a clump of cells to an organism with a certain function can occur is really something that we can now try to explore. And with that, I'm just going to maybe just advertise for this. If you are interested in some of these ideas, ideas in active matter. This was a perspective we wrote about 2 years ago after a KITP meeting with my colleagues Christina Marchetti, Mark Bovik, and Sriram Ramaswamy. And this is my amazing group. This is a recent picture, but these are all the amazing people within my group and, of course, my collaborators across the globe. And thank you again for having me.